Hello class, this is Mr. Mormon. Welcome to video number 4 in our unit on cells. Today we're going to be learning how to make wet mount slides. We're going to learn about staining techniques, and we're going to learn how to measure using a microscope. So let's start with how to prepare a wet mount slide. The first thing you need to do is obtain a clean microscope slide and a cover slip. A cover slip is a very thin piece of plastic or glass which goes over the specimen. It permits the objective lens to be lowered very close to the specimen. Next, you're going to place the specimen in the middle of the microscope slide. The specimen must be thin enough for light to pass through it. Next, using a dropper pipette or eyedropper, place a drop of water on the specimen. Next, you're going to lower one edge of the cover slip so that it touches the side of the drop of water at about a 45-degree angle. The water will spread evenly along the edge of the cover slip. Using a dissecting needle or probe, you're slowly going to lower the cover slip over the specimen and water as shown in the drawing. Try not to trap any air bubbles under the cover slip. If air bubbles are present, gently tap the surface of the cover slip over the air bubble with a pencil eraser. This will move the bubble to the edge of the cover slip and it will be out of your way. And finally, remove any excess water at the edge of the cover slip with a paper towel. If the specimen begins to dry out, you can add a drop of water to the edge of the cover slip. So here we see a student carefully following the procedures for making a wet mount slide. The specimen was placed in the middle of the clean slide and a drop of water was placed on top. The cover slip, which you see here, is being placed down at a 45-degree angle. Again, this is to eliminate air bubbles as much as possible. Carefully lowering the cover slip will push the air out to the side. Finally, excess water can be removed with a piece of paper towel at the edge of the cover slip. Okay, now let's talk about staining. Sometimes we'll add stain to our specimens so the cells will be easier to see. Stain is like a dye which colors the cells, making them stand out better and making them easier to be seen under the microscope. First, you'll follow all the same procedures we just went over to make a wet mount slide. You're then going to add a drop of stain at the edge of the cover slip. Using forceps, touch a small piece of lens paper or paper towel to the opposite edge of the cover slip as shown in the drawing. The paper causes the stain to be drawn under the cover slip and to stain the cells in the specimen. Okay, next we're going to talk about how we measure with a microscope. And to understand how we do that, you need to understand what the field of view is. The field of view is the area that you can see under the microscope. Under low power, the field of view is larger. You see a larger field of view under low power. Therefore, the cells are going to appear a little smaller. When you go to a higher power, the field of view gets smaller. You see less area, but therefore the cells, of course, are going to look bigger. To measure the field of view, you're going to place a thin, flexible, transparent ruler like you see here under the microscope and focus in on the metric edge where each one of these little lines represents 1 millimeter. The space between the lines is 1 millimeter. You can also use a grid slide which has this little grid pattern on it. Each one of the boxes of the grid is 1 millimeter. Using these tools, you can measure the distance across the field of view in millimeters. We'll measure in millimeters, but very often cells are expressed in micrometers. 1 millimeter equals 1,000 micrometers. And since there's 1,000 millimeters in a meter, that would be 1,000,000 micrometers in a meter. In other words, a micrometer, or micron, as they're often called, is 1/1,000,000th of a meter. And this is the symbol for micron or micrometer. It's kind of like a backwards U with this tail on it. Okay, so in this example you can see what I'm talking about. The metric ruler has been placed under the microscope, and this is what we see when we focus on the edge of the ruler. We can now measure the field of view is 2 millimeters. Again, each one of these spaces between the lines is 1 millimeter, so that's 2 millimeters across. Once you've measured the field of view, you can now start estimating the size of objects in the field of view. Here we see both the specimen and the edge of the ruler under the microscope. We can estimate the field of view. Again, if this is 1 millimeter, the field of view might be 1.3 millimeters. And the specimen, which looks like it's about maybe halfway across the field of view, might be estimated at 0.6 millimeters. Okay, let's do some other examples. The diagram below represents the field of view of the low power objective of a microscope. Each dark line is a millimeter line on a metric ruler. What would you estimate the field of view to be? Counting the spaces, I get 1, 2, 3, 4, 5 millimeters. Now in this example, you can see what it would look like when you put a grid slide under the microscope and look at the field of view at different powers. In this example, you need to estimate the field of view based on what you're seeing from the grid slide. Now remember, each one of these boxes is 1 millimeter. You're not going to count all the boxes because that would give you the area. What we want is the distance across. In these examples, you have to estimate to the nearest 0.5 millimeters. So what would be your answer for this first one? If you count the boxes across, 1, 2, 3, 4 millimeters, maybe a little more than that, but to the nearest 0.5 millimeters, I would get 4.0. This next one looks like 2 whole boxes across, and then part of these other boxes, maybe 2.5. This next one is a little more than 2.5, I'd say, but to the nearest 0.5 would be, again, 2.5, I would estimate. And this last one is 1 millimeter plus this portion, maybe to the nearest 0.5 would be 1.5. Okay, in this example, it's saying that this is 1 millimeter from this line to this line. So each one of these smaller divisions would be 1/10 of a millimeter. How long would you say each one of these cells is? What's the distance across the nuclei? I get 0.5 for the length of the cell and 0.1 for the diameter of the nuclei. Okay, in these examples, they give you the field of view. You have to estimate the size of the creature to the nearest 0.1 millimeters. This first one seems to be a little bit less than half of the field of view. So half the field of view would be 1 millimeter, so maybe 0.8 millimeters. On this next one, if it's 2 millimeters across, this one might be 1.5 millimeters in length. These guys, if you include their tail or flagella, they're about, halfway across the field of view, maybe 1.2 or 1.3 millimeters. And these last ones, it looks like you could fit about 3 of them across the field of view, which would make each one about 0.5 millimeters. And here's our last problem. They show you the edge of the ruler, what 1 millimeter is. How big would you estimate the size of that organism? I get 0.5 millimeters. It's about halfway across the space between the 1 millimeter lines. All right, that's it for this video. If you have any questions, of course, ask me tomorrow.